Morgan, thank you so much for joining. It's been really yeah, quite fantastic well, I'm very proud of myself for having been able to get you onto this call, because your name has definitely fallen more than twice. I think in this group we've been talking a lot about biomarkers lately. In fact, I think maybe even Ravi is on this call, who has been kind of like spinning off a side project on biomarkers, which is a separate group that is now meeting. But it's been of great interest to this group and on a variety of different levels. I think they can tell you best. I won't try to be the soundboard for this. But thank you so much for joining. I discovered your research really kind of as a coincidence as I was kind of like scrolling the web for different ways of how one may be able to do biomarker standardization. And I found the fantastic paper that you co-authored, which I'm going to post here as well on. Which you co-authored really quite recently, and it was on underlying features of epigenetic aging clocks. I'm going to post all the links to your bio and to the paper in this chat. But for now, I want to give the stage all up to you. I think you're going to talk for about 30 minutes. I'm going to try to collect questions in the group. I'm already going to ask folks to please collect questions marked with a Q in the group, and then we can maybe streamline them in for a discussion afterwards. Thank you so much for joining. I can't wait to hear what you are about to share. I am going to make you co-host right away so that you can share the screen. All right, Morgan, take it away. It's really a pleasure to have you here.
All right, thanks so much, Allison. I'm excited to be here. I see a lot of familiar faces, so this will be good. And yeah, I'm excited to share this paper that I think came out earlier this year, I believe. So it's underlying features of epigenetic clocks. The actual is in vivo and in vitro. I didn't put that on the title. So I usually, when I'm not giving a talk to aging researcher, I start with the idea, aging is the biggest risk factor for death and disease. And we think that this is actually causal. So the things that are changing with aging are actually causal in disease pathogenesis. And for this reason, we really want to quantify aging beyond just a proxy of using chronological age. So aging is actually something very easy to perceive. My 5-year-old can distinguish an older individual from a younger individual. Oh, hold on, let's see if it'll go forward. There you go. And that's because it impacts nearly every cellular system in our body. And this explains why the consequences of aging are so dire, right? It impacts, or at least drives, a bunch of different diverse diseases. But this also is an issue in that it's not exactly clear how we would quantify aging. So I almost think of this as like you go to a Starbucks and there's almost too many options. So it's not exactly clear where to start. So people have started to use, you could use images of someone's hand to actually make an aging marker. You could use facial images, essentially anything. But the hard thing is to actually determine which information is the most important. If the long-term determination is figuring out who is at risk for death and disease, and also if then intervening and actually seeing a change in that biomarker, we believe should impact differences in health span and lifespan. So my lab likes to approach this kind of from a systems biology perspective, in that we start to think that aging probably starts at the molecular level, and this then manifests upward to higher levels of biological organization. So, you have changes at the molecular level, then the macromolecular, organelle, cell, tissue, organ, system, and eventually the organismal level. And so failure at each of these lower levels will then impact aging phenotypes at the levels above. So we study aging at the kind of molecular, macromolecular level. And specifically, we work on epigenetic clocks, which I'm sure a lot of people in this meeting are familiar with. But if you're not, this is mainly, even though we call them epigenetic clocks, we really are focusing specifically on DNA methylation. So DNA methylation is actually the most abundant epigenetic modification in eukaryotic cells. And it really influences kind of the chromatin structure. So whether you have heterochromatin versus euchromatin and kind of how it's packed. And DNA methylation is really important because it determines cell identity, it determines cell phenotype, and basically how every cell is responding to its environment. So I like to use the analogy, I know there's a lot of analogies for epigenetics or DNA methylation, but I like to use the analogy of a recipe. So basically your genes are all the ingredients that you need to make anything, and your epigenetics or DNA methylation is the recipe on how each cell is going to make whatever it's going to go about doing. And essentially what we know is that with age, this recipe gets a little messed up. So maybe you add too much of something, or you completely don't add a different ingredient. And this really is going to change the overall phenotype of the cell. So when we're looking at this data to actually describe what this looks like is we measure DNA methylation at what are called CpG sites, so CpG dinucleotides. And so these cytosines can become methylated, and we measure this essentially as a number between 0 or 1. So in your population of cells, what proportion of this exact cytosine are methylated versus unmethylated. And we see really interesting changes at very specific CpG sites with age. So for instance, perhaps in this cytosine, if I looked at a sample from a 20-year-old, perhaps you would have 90% of cytosines at this location would be methylated. Versus if I looked at an 80-year-old, maybe that drops to 60%. There are other regions that you get increased methylation with age. So this isn't a single direction. So some CpG sites increase with age, some decrease. So for this example at the bottom, in a 20-year-old, perhaps you would have 5% of cells methylated at this location versus in an 80-year-old, you get 45%. These two represent what we would call drift. So they're moving towards 50% from the two extremes. But you also get some interesting ones which actually are also moving away. So you start perhaps as a 20-year-old with 20%, and then by the time you look in an 8-year-old, it's only 1%. So because these changes are actually so precise, and actually interestingly, even precise across different tissue and cell types, people have developed what are called these epigenetic clocks. So basically what we can do is we can take a blood sample, a tissue sample, or even cells in culture, and we'll sequence DNA methylation and get information on between 20,000 to oftentimes millions of CpG sites across the genome. And then people have applied different supervised machine learning approaches to actually train predictors of various aging outcomes. Usually we're training predictors of chronological age, although the newer clocks have been training predictors of what we might call age correlates or phenotypes of aging. And then what we get is your predicted age based on some subset of these CpG sites, and you can compare those to the observed age of the individual or the sample and ask if kind of the discrepancy between that is predictive of, for instance, mortality risk, morbidity risk, or any other outcome of aging that you're interested in. So dozens of these epigenetic clocks have been developed. So the first one was developed in 2011 by Bocklint et al. But since then, there's been a ton of these, and actually I don't even have all of them on this timeline because there's so many and I can't even really keep track of them. A new one seems to be coming out every month. So kind of these ones in the early, that were developed earlier, what we might consider these first generation clocks. So again, these were trained to predict chronological age, whether in blood or in multiple tissues. However, some newer ones, like the one we did in 2018, the Grimm age clock, the Belsky pace of aging clock, are what we might consider second generation. So these weren't trained as chronological age predictors, but rather predictors of things like mortality or some phenotypic age or other measures related to that. And today, also at the very end, I'll talk about the meta clock, which is a new clock that's part of this paper. So one interesting thing, as we started to look at these epigenetic clocks is we started to ask, they're all attempting to approximate the same signal. Like we're all trying to capture what's happening to the aging methylome. and understand how that, either the mechanisms that feed into that or also the downstream consequences. So I like to think of this as there's, a tutorial to draw a hand. And even though these are all meant to actually be approximations of this hand, they obviously look a little different. So some are a little bit better than others, some a little worse. And the epigenetic clocks are, you can kind of think of them along the same lines. They're all attempting to capture the same signal, but they're not actually getting the same exact thing. So yeah, they're trying to approximate the same signal, but you actually get fairly different results when you compare the different clocks. And that's because they differ either in terms of the training sample characteristics, so who was included in that training sample when they developed it, the outcomes they were trying to predict, whether it was chronological age or some of these age correlates. Some of them were trained in whole, actually a majority of them were trained in whole blood, but some also incorporated other tissue types. And some of them actually had this pre-selection criteria. So they didn't look at all the CpGs that were sequenced, but really focused in on certain types of CpGs. So you end up with slightly different clocks. And what we were asking is, what do the different clocks capture? Are there some shared signals across them? Which ones are better at approximating specific processes? So this is the paper that I'm going to discuss today. The first author was one of my former postdocs, Zuyun, who's now moved on and has a faculty position of his own. And basically what we did is we had this kind of hypothesis development where we compared 11 of these epigenetic clocks in terms of their tissue prediction, their association with gene expression, and also differences in terms of tumor versus normal tissue, and also in vitro aging hallmarks. Then what we did is we actually deconstructed the clocks. So we tried to determine, if you imagine a Venn diagram, not in terms of the CpGs in them, but if you think of it more abstractly like a signal, are there some signals shared that are core across the clocks and some that are different? And by knowing that, can you actually put them together to make an even better clock than any of the originals were? So for the first part, just comparing the age predictions across tissues, what you can see is that different clocks have very different age predictions. So this is the kind of famous Horvath clock, the original pan tissue clock. You can see it has the highest age correlation. And that's because it was trained as a multi-tissue age predictor. But the other interesting thing is actually a lot of these blood-specific clocks, or blocks that are trained in blood, actually show very good age prediction, or at least age correlation within other tissues. So there's some kind of signal that is not blood-specific that they're capturing, but also tracks across whether you're looking at fibroblasts or whether you're looking at samples from brain. But also when you look at these blood-trained clocks, you'll notice that they seem to suggest that tissues are actually aging at different rates. And we think that actually the Horvath findings that show all tissues are aging at the same rates is actually an artifact of the method that was used to develop it. So it's actually, if anything, adjusting out tissue differences so that it gets consistent age predictions across. And actually what we would expect would be that tissues age at different rates. We know they have different kind of different environments, they have different replication rates. So it's not surprising. And also what's pretty consistent across these other clocks is that it seems that brain is aging at a slower rate than most of the other tissue or cell types. So the next thing that we did was so a lot of people had taken the genes in which these CpG sites are co-located in and run either a pathway enrichment analysis. But what we did is we actually just looked at differential expression associated with the epigenetic clocks. And we didn't want to capture something that was specific, again, to one tissue or cell type, because the epigenetic clocks are fairly universal across tissues and cells. So what we did is we took gene expression data from purified monocytes and also bulk cells from dorsolateral prefrontal cortex. And we asked whether there are conserved transcriptional changes that were associated with either accelerated or decelerated epigenetic aging. And we used a network analysis to do this. I won't go into the methods, but I'm happy to answer questions if people are interested. But first, we just asked whether the clocks seem to have similar associations with transcripts. And basically, this shows that at least this core set of clocks, so Yang, Hannum, Lin, Levine, and the 2 Horvath clocks, have very similar associations with gene expression. This is in monocytes and also in brain. So they seem to have shared transcriptional signals. This also shows the strength of those associations. So when this is relative to the original Horvath clock, when the slope is greater than 1, that means that clock actually has stronger but similar signals. So for blood, the Hannum clock, which is not surprising, it's a blood specific clock, had the strongest transcriptional signals. And in brain, actually this Horvath 2, which is the skin and blood clock, actually had really strong signals compared to the original Horvath tissue clock. So when we actually looked at what types of genes were either associated with accelerated or decelerated epigenetic aging, again, we use a network analysis, so we aren't looking at specific transcripts, but instead groups of transcripts. What we find is that higher epigenetic age was associated with a decrease in expression for genes associated with things like cellular respiration, mitochondrial translation, oxidative phosphorylation, and also a decrease, again, in kind of these mitochondrial gene expression for this, what's called this green module and this turquoise module. However, we also found that they're associated with an increase in genes associated with kind of chromatin modifications, histone modifications, and also kind of cell cycle checkpoints and potentially autophagy down in this red module. So next, we compared whether, so a question I get a lot of times is cancer or tumors actually accelerate in their epigenetic age versus normal? And the answer is actually depends on which clock you're looking at. So you get fairly different answers. So at least we found for our recent, the pheno-age clock, we get significantly increased epigenetic age in tumor versus normal tissue across 4 tissue types. So breast, colon, lung, pancreas. Same was true for the Yang clock, which is actually a mitotic clock. So it's supposed to capture kind of mitotic history. But for the other clocks, we actually find no significant difference between tumor or normal. And in fact, in the original Horvath clock, we actually find a slight decrease in tumor versus normal, which again, we think is a function of how statistically it's adjusting out some of the signal for replication. We also looked at data from cells in culture. So for this, we had data where we actually induce cellular senescence. So we do it using 2 different induction methods. So one is serial passaging, where we either passage cells to become replicatively senescent, which is shown in blue, or what you would call near senescent, where they're still proliferative, but they're expressing high senescence associated beta gal. And then also looking at cells induced via oncogene, so HRAS retrovirus, and we can compare these to the early passage cells. And basically what you can see here is, again, the pheno-age clock does show an increase for the near senescent and the replicative senescent compared to the early passage, and a slight increase for the oncogene-induced senescent cells compared to the early passage. Same thing for the Yang clock again, although it was not significant for the near senescent. And then interestingly, the Hannum clock does not pick up replicative senescence, but it did actually have the strongest effect for the oncogene-induced senescence. And the Lin clock picked up only replicative senescence, but not oncogene-induced senescence. And neither of the 2 Horvath clocks significantly showed any significant change as a function of senescence. We also had a perhaps imperfect model of mitochondrial dysfunction, basically where we have depleted mitochondrial DNA, so these row 0 cells. And again, we show that the Levine, the pheno-age clock, the Lin clock, and the Yang clock show an increase in epigenetic age in these row 0 cells compared to the controls. There's a slight increase for the 2 Horvath clocks, however, it wasn't significant. I believe there's only 3 samples in each of these categories. So the next question is, what we find is that the epigenetic clocks are actually behaving slightly differently depending on if we're looking at age correlations across tissues, whether we're looking at cells and culture. And we didn't show it in this paper, but a lot of people have also published differences in terms of their predictions of different diseases or things like mortality. And what we actually think is that the epigenetic clocks are composites of a lot of things that are changing in the methylome with age, and that these different parts are actually mapping on to different aging processes. And so each clock is going to either prioritize certain parts or be enriched for certain parts. And by actually decomposing the clocks into these specific pieces, we can determine more mechanistically what the different clocks are capturing, why they behave slightly differently in terms of their predictions, and also which of these pieces actually are most important for outcomes of aging that we care about. And finally, I think where the field is going is trying to determine, are all of these parts modifiable? So are all the epigenetic age changes modifiable, or is it only certain ones that we can target? Ideally, what we would want to find is that they are modifiable, and the ones that are modifiable are the ones that are actually predicting disease or mortality outcomes. But I think you can't do that when you look at the clocks as a whole, because again, they're these composite measures. So again, we use what's called a consensus network analysis. I again won't go too much into this. We've actually redone it since that paper, so I'm showing our improved version that my postdoc Albert Higgins-Chen actually took over. And so what we do is we look at all the CpGs across all the clocks. So we've now expanded this to include additional clocks, and we can then put the CpGs into what we call either different modules or different networks. So the Horvath clock has 353 CpGs, and we'll assign each of those CpGs to a different module based on co-methylation across a variety of different tissues. And then what we can see, so this up here is signifying which clock it's found in. So if you look, this is the original Horvath clock, and you can see it has some CpGs that are assigned to this yellow module, some in the green, some in the turquoise. And down below are the age correlations for these CpGs in different tissues. So the ones with this kind of blue box are ones that tend to decrease methylation with age, and the ones in the red box are ones that increase methylation with age. And you can see that there's some distribution across clocks, although there are certain clocks that tend to really capture very specific processes. So I'll go a little bit more into kind of what those are. So again, as I mentioned, every CpG in each clock gets assigned to each of these modules. So then we can say, are some clocks enriched for certain modules, and is that why they behave slightly differently? So what you can see here is, so this is not all the clocks, but a majority of them, and they do actually have fairly different distributions of these modules. So, the Horvath 1 clock has a big enrichment for this turquoise module, whereas Grimage essentially has very little turquoise module, but a much bigger enrichment for gray or blue. And then we can also ask the question, even if they all have the blue module, because every clock will weight the CpG slightly differently, are the blue modules actually capturing the same thing? So when we look at the turquoise, we find very high correlation. So they're all capturing the same signal from that set of turquoise CpGs. So even though they don't have the same CpGs in the clocks, in each of the clocks, they're capturing that signal by probably using clocks that are CpGs that are highly correlated. But when you look at the blue module, they actually have pretty, a lot of discordance in the signal that they're capturing from this. So the next question is, which ones are actually capturing the correct one, and which of these modules are the most important for outcomes that we care about? So what we did is we actually used these different subclocks to train a mortality predictor. And then we compared that to at least the original pheno-age clock and also the Jean clock, which is a mortality clock. And we used the Framingham data for this. And what we can find is this new, what we call meta clock, which takes pieces from each of the different epigenetic clocks to create what we think is a better clock, actually has a much better predictor of mortality than either of these two. So the Levine clock, for every 1 standard deviation increase in your epigenetic age relative to chronological age, you have about a 2.6-fold increased risk in mortality. Whereas for the meta clock, this actually ends up being a 5.6-fold increased risk in mortality for every 1 standard deviation increase in the clock. We also show that even though this is trained as a mortality predictor, it actually tracks really strongly with age in a variety of different tissues. So we looked at whole blood, breast, dorsolateral, prefrontal cortex, so brain and dermis and epidermis. And again, we find that this is tracking pretty strongly. Again, it's not trained to be an age predictor, but still captures age. And I'm not showing this here, but you actually, in epidermis and dermis, you get slight differences, but it also captures whether it's taken from a sun-exposed versus an unsun-exposed region. Again, it captures the kind of mitochondrial DNA depletion and cellular senescence for both oncogene-induced and replicative senescence. And then finally, I wanted to quickly show this. This is where we're moving with this. So it's not actually in the paper, but I'll just show it really quickly. Is basically you can now look at these different module clocks and ask which ones are tracking which mechanisms. So I'm not going to show, we're doing some stuff with replication, some stuff with senescence, some stuff with mitochondrial dysfunction. And you can see some of these modules track better with age, but something that we're really interested in is kind of this epigenetic reprogramming, and if all of the clocks are equally responsive to epigenetic reprogramming. So this is a time course, basically, where you start with your somatic cells. I believe this is fibroblasts here. And then you, using Yamanaka factors, I believe this is 5 factors, but I'll look back and see if people are actually interested. You can track changes in these specific modules. And what we would expect to see would be them to go down, right? You want, this isn't an absolute value of age, but you want it to decrease in its relative epigenetic age. So at this stage, these cells are partially reprogrammed. So for people who are following kind of this partial reprogramming, this is kind of the sweet spot of where people think that we want to be hitting. You want to do kind of this pulsed reprogramming, and you don't want to de-differentiate the cells, but you actually want to try and reduce the age signature. And then you get incomplete, and then finally fully de-differentiated induced pluripotent stem cells here. And again, what you can see is not all the modules or not all the types of epigenetic age measures are responding to this. So there are a few, for instance, the blue module, salmon, brown, and red, that seem to actually show a stronger effect for reprogramming. However, there's some that actually increase their quote unquote epigenetic age as a function of reprogramming. And then the one other thing is we actually went back and looked at some data on reprogramming that had been published. So this is from the original paper that was published, where they used the Horvath clock and showed that this partial reprogramming, they can get a decrease in epigenetic age in fibroblasts and endothelial cells. And this is the exact same data, just plotted on a spiderweb plot. So basically the ones in blue are the reprogrammed cells, and the ones in red are the fibroblast controls. And the closer the dot is to the outside, the older it is. So you want your reprogrammed cells to all be around the middle. So this is showing the exact same thing. But when we actually parse it out by modules, we find that it's completely captured by just a piece of the Horvath clock. The whole clock is actually not being reprogrammed. It's just this one module. And so the question then becomes, you know, is that module, what mechanistically is that capturing? And is that one that we actually care about in terms of predicting future mortality or health measures? And it's still early and we're looking to this, but actually that does seem to be the most predictive of things like all-cause mortality. And I'm not showing that data because we're just starting to look at it. But just in conclusion, basically epigenetic clocks have some shared transcriptional associations that suggest there might be some links between epigenetic aging and things like metabolism, immunity, and autophagy. Only 2 of the clocks, Levine and Lin, reflected both the hallmarks of aging we looked at, so cellular senescence, either from replicative or oncogene-induced, and also mitochondrial dysfunction. Using network analysis and ensemble learning, we're able to identify unique signals that are underlying some of these epigenetic clocks, and in the future, hopefully look mechanistically at what those are capturing. And we can also use these modules as input to train potentially better epigenetic clocks going forward. So you might combine certain pieces if your goal is to predict cancer risk, or maybe a different piece if your goal is to predict Alzheimer's disease or whatever it may be. But actually, by using these different pieces and understanding how they map to different outcomes, we think that we can predict better clocks. So this meta clock, which we showed in this paper, is a better mortality predictor than some of the prior existing epigenetic clocks, but also was applicable to non-blood samples and seemed to also capture things in vitro. So with that, I just want to thank the people in my lab. This is an outdated pre-COVID picture, so a lot of people aren't pictured, and also some of my collaborators and my funding from NIA and Glenn. I'm happy to take questions and discuss.
Thank you so much. Fantastic. I'll clap like this. I think I can see a few hands already up. Okay, so we have a bunch of questions already collected. Thank you so much for sharing. So for new data and or at least a new interpretation of already. It's very helpful. Okay, I'm trying to go chronologically. I think that's to everyone who's in the chat. If you want to upvote questions, then feel free to do so. Otherwise, I'm going to go chronologically up. The way to upvote them is just
Alison, you don't go chronologically. You have to go biologically. I'm sorry, you didn't learn the lesson here.
Oh my God, we need a good clock for that. So a good clock for the chat would then be the upvoting and downvoting. That let's do that as a standardization. I think Creon had the first one there, and you can moderate the questions up as you so choose. Creon, go for it.
Okay, yeah, mine might be a little too general, and so maybe it should get down moderated. But what I want to know is, can you maybe up front just summarize what the purpose of clock research is? Is it to, I mean, it's obviously not to predict someone's chronological age or even the chronological age of their tissues, so what is the underlying or purposes of this research as you see it?
Yeah, so that's an important question. And I imagine that different people have different answers to that. So actually some people might say that it is to predict chronological age. So I know there are some of these applications for forensic research or even studying animals in the wild. If you can actually tell someone this is whatever age animal. So it isn't necessarily not to do that. So in some applications it is. At least in terms of the way I think that it's going to be the most useful is, number one is I actually think it will inform some understanding of mechanism, right? If we can link molecular changes to kind of later health outcomes and then also link those to different mechanisms, that might be an easy way to do it. Two, I actually think they'll be potentially very useful for intervention trials or clinical trials that don't have 10 to 20 or 30 years to wait to see if the intervention group dies later than the controls. And third, I think, I actually think they'll be very useful for kind of preventative medicine. So secondary, primary prevention to actually understand individuals' risk of death and disease much earlier in the process, and potentially even inform kind of, I'm not saying necessarily health choices, but actually once we get further along with this, potentially informing individual health choices and also kind of medical care as well.
Thanks a lot. Let's cede the floor.
Lovely, thank you. Okay, next one up, we have Tom. Tom, can you say whether your initial questions were to Morgan or whether they were just for the group at large? And maybe Morgan can help answer them anyways.
Yeah, Morgan, one question I had is whether there is data that could be from cohort studies or biobanks that would be useful to the aging research community that you don't currently have access to that you would like to have access to.
So to clarify, is that there are samples or existing data?
Both.
Okay, I would say samples. I mean, I think the fact that UK Biobank doesn't have methylation is very disappointing, at least to me, and I think a lot of other people, because they have a wealth of other data that would be great to link to this. I think a lot of the cohort studies are generating methylation data. So Framingham, Women's Health Initiative, the Health and Retirement Study, I heard NHANES is getting methylation data soon. But I think the other thing that's lacking is longitudinal data. So there's very few studies that have collected this longitudinally, so we can actually understand within individuals how these patterns are changing. And ideally, what I think in the future you would want is not just a general measure for everyone, but I would want, for me, compared to me 10 years ago, how have I diverged and in which way, and what does that mean for my future health? So I think the only way to do that is to have longitudinal data where you can put an individual in reference to themselves rather than the kind of population as a whole.
All right, thank you. Did that answer the question? I think so, Tom. Yes? Okay,
Yep.
Great, lovely. Next one up, we have
Oops, there we go. Hey, so I guess my question is, can you make a clock that uses a blood sample to detect your liver failure or your Alzheimer's disease or whatever, which is clearly something that you're like getting at, right? But if we're going to use it in trials, we're not going to do brain biopsies. Yes. So is there any, I'm sure some people would have published that if it existed, but are people pushing towards that, I guess is the main question. And maybe also what do you see as the feasibility or main challenges there?
Yeah, so we're actually working on that exact question. And obviously some of these are going to be easier. So it does seem like you can develop a clock that's going to capture liver, I mean, maybe not predict liver failure, but it captures something about liver aging. Brain seems to be a little trickier. Obviously we can make really good clocks using brain samples, but as you said, you're not going to take brain biopsies. So we are actually working on whether you can use blood, saliva, or buccal, or some other easily accessible sample to get an estimate of kind of brain aging. The signal is not as straightforward, and the question is whether you're going to need to use cell-free DNA or actually if you can capture it using kind of leukocytes. So yeah, to be determined, but yeah, I think that's an important thing that we agree.
So it's not clear yet whether, like, let's say the liver works better. Is that because liver secretes a bunch of stuff and so the blood cells are more exposed to the results of the liver's dysfunction versus cell-free, you're picking up actual liver DNA?
No, I think it is that you can actually capture it using blood cells, so leukocyte DNA methylation, because I think of this exposure to liver proteins from liver and metabolites and lipids and stuff. Yeah. Exactly.
All right, thanks.
Mm-hmm.
All right, Ravelar Vargas, you're next.
Hi, yeah, I'm just wondering, basically the biggest risk factor for cancer development is aging, but not all cancers are aging related. And some types like breast cancer actually decrease after specific ages. So I'm just wondering how these clocks might diverge based on the type of cancer.
So we've looked at a few cancers. So we have looked at breast cancer. They are predictive of future incidence of breast cancer when measured in blood, and actually probably more so when measured in breast. We've looked at normal tissue from women who have had cancer versus those who have not, and the clocks are accelerated, even though we're looking at normal tissue. I would argue that, yes, not every cancer is age associated, but even some of these that show a decline later, I think is more of a selection. So people who are at, Nir can probably answer this. There is this resilient subgroup of individuals who seem to be at much less risk. And when they're the only ones left in the population, it's going to look like an artificial decrease in the risk of some of these diseases. But I think it's a selection process. So I actually think that might not map on to whether the clocks are going to pick up breast cancer or other cancer risks. We're really interested in how mitotic rates in different tissue types maps onto epigenetic aging, and then how that relates to cancer risk in those different tissues. But that's it's fairly preliminary. We have some data, but not enough to make a strong comment on that yet. I hope that answered the question.
Thank you. Next one up, we have Ravi. And Ravi, maybe you can say a word or two in case Morgan doesn't know you, just about who you are, your background, why the question is relevant, then she has some context.
Yeah, Ravi Pandey. I work in Microsoft Research in a partnership with Adaptive Biotech on immunosequencing and trying to understand that, as well as the background in genomics and cancer genomics. So my question, and I'm also helping lead some discussion within this group around biomarkers and trying to figure out if there's an interesting project that we can pull together related to biomarkers. So this is really relevant. The one question I had was sort of looking at these different modules. Have you sort of gotten some sense of if they're functionally different and what those functions are that affect those different modules? And then, are they causal, are they upstream or downstream of the things that actually have the phenotypic effect, and how do they maybe relate to different interventions?
Yeah, so that's what we're working on right now. We have a little bit of data on that. I didn't show it for the sake of time and because it's very preliminary, but the idea is to map the different modules to functional mechanisms. So whether some of them are picking up, so it seems like some of them are changes that are happening as a function of cellular replication, and understanding kind of why and what those are and what that means. Some of them are probably picking up things like cellular senescence, so like a change in the cell state. There could be a number of other things, pluripotency or stemness of the cell. So yeah, the goal is to understand, to map these to different mechanisms, which we think you can't do looking at the whole clock. You have to kind of piece it apart and say, this mechanism is mapping to this part. And once we have that map, say which things are causal or upstream or downstream of different things, and which things are driving different kind of phenotypic manifestations of aging that we see. So that's kind of the long-term goal, and we're a little bit early doing that, but they do seem to start to map to different mechanisms. When we're looking at them, we're doing a lot of work in culture.
Great.
Lovely. Okay, next one up, we have Lee, and then Aubrey with a question I think that's directly relevant to this, so maybe we'll stay on topic. Lee, also 1 or 2 sentences about who you are would be awesome.
Well, I specialize in the quantification of health, wellness, and aging. I think I'll leave it there at that. I have a business coming out in the next 2 months, which will be based upon that. Morgan, I would like to ask you, how would you describe the Gen 2 and the MetaClock in terms of being responsive to interventions like lifestyle, like nicotinamide riboside, supplements, diet, and so forth?
So I think that's the outstanding question, is we don't know whether these are modifiable or what interventions would modify them. I think we do know, I mean, we have some indication that they are probably modifiable, right? Because you can look and say, well, we've looked at GWAS or twin studies, and it only explains a very small proportion of the differences between individuals and their epigenetic aging. And you can also look at observational studies at behaviors and say, you know, all the things that your mother told you to do seem to be associated with better or lower epigenetic age. So we do think that lifestyle things, I'm not saying supplements, but our behaviors, whether it's exercise, nutrition, sleep, stress, do change the epigenetic aging. We don't know the degree, and I would also argue it's probably going to be different for every person, right? So, you know, if I ran 30 minutes a day every day, that might have a different effect than if you did. And I think that's where we don't quite understand this kind of personalized.
But do you think you'll be able to pick it up in clocks? Like
I think we will be able to pick up some, again, based on the observational studies. But again, I would not suggest that epigenetic clocks are the only biomarker of aging we should be measuring or the only indicator of health. They're going to capture, I think, alone capture a fairly good part of the signal, but they're not the whole picture.
Do you have any thoughts on transcriptomic or proteomic clocks?
Yeah, so I'm really excited about the proteomic clocks, and we're actually doing work, some stuff with NIR, some stuff with people like Luigi Ferrucci on trying to understand integration there. The transcriptional clocks, I think, are a little bit noisier, but I don't think that means that they're not useful or informative. The one thing that makes me excited about epigenetic clocks in relative to the other types is that they seem to be universal across most tissue and cell types, whereas the transcriptional proteomic clocks seem to be a little more tissue or cell specific. So that just interested me from a basic science perspective, not necessarily in terms of application. Just understanding what it is about this kind of universal aging change that happens regardless of cell or tissue type that you look at. But yeah, I think that's a long way of saying, I think the transcriptional and proteomic clocks are also useful and informative. I might not say the same thing about telomere length, but yeah, but there's definitely other useful clocks out there.
Thank you.
Lovely. Aubrey, your question.
Yes, sure. Hey, Morgan, that was a lovely presentation. Thank you for joining this group. So as you were quite right, of course, in mentioning that there are diverse reasons for interest in such clocks, things like forensics, but I think it's fair to say that the broadest interest is in the prediction of interventions to postpone chronic problems of late life. So of course, the big question is, how can we make a clock that is a really good predictor, given that what we're interested in are new interventions, which by definition, the clock has not been trained on. So I'm wondering where the thinking is on this right now. And particularly, I'm interested in the possibility that we might take existing interventions, ones that both work and ones that don't work, and do like measurements of epigenetic age or whatever, biological age, before and after, and try to to get as broad a correlation as possible. But I haven't seen much discussion of that. Am I making sense?
Yes, no, I mean, this is very much what we're thinking is, I would not say that any of the existing epigenetic clock measures are the ones that probably should in the future be used for these interventions. I think we need to integrate some of the data on the interventions to actually improve our clocks, right? So one way to do this would be if you did have a huge human trial like TAME or some other trial where you have methylation measured, before and in the middle, and then you have these outcomes, is you can actually train that to figure out what methylation patterns are capturing, assuming, metformin or whatever it was actually does decrease incidence of disease. You can actually then train what methylation patterns are capturing that difference in incidence and disease in the treated versus control. But the other thing, I have a grant with Vadim Gladyshev that's looking, at least in mice, at interventions to try and understand kind of the change in the methylation patterns as a function of the interventions, which I think is a little bit more what you were getting at. The other thing I'm interested in related to that is, calorie restriction has kind of different effects on lifespan in different studies and in different animals. So can you actually figure out, is there a methylation pattern that's predicting the effect or kind of the benefit of caloric restriction? And I actually think those measures are going to be more kind of informative going forward, whether you're looking for a calorie restriction mimetic in the future or something related to that. But I think we need to use all the intervention data that we have to actually improve the measures, so almost work backwards in some regard.
Allison said I'm up next. So, but before I get to my actual question, that, for the nice thing about mice is you can actually then also follow them up till they die. And, so, I mean, that would be great, right? Is do more of this stuff systematically in mice. I'd also love to see an analysis like your papers that looked at the differences in terms of training models differently on which you would need to do in mice, training models differently in terms of time till death at different sort of durations, right? Different time, what's different between a clock that's predictive X time out versus predictive 2X time out? That would be really interesting to look at. Well, my actual question is, are you involved at all with Steve Horvath's efforts to try and get any kind of official FDA blessing on some version of a methylome clock for some purpose? Last week in this group, we talked a little about the huge gap between a biomarker, which is useful for research, and a full-on surrogate endpoint for a clinical trial. And I think there's sort of broad agreement that we're not at the surrogate endpoint level. And I suggested that there's an in-between point here because, many indications that people run clinical trials for are for conditions where age is a risk factor, and therefore a normal process of running the clinical trial is that you try to balance the treatment and control by age groups, and an inevitable part of the analysis is to stratify by age when analyzing efficacy. Do you think that methylome clocks are already at the point where they would actually be better than using chronological age in terms of efficacy analysis, in terms of what age version you're using for stratified analysis?
Yeah, I mean, I think that is another important application that they could probably even sooner than as an endpoint be used in some of these clinical trials. We're also seeing in some of these smaller clinical trials that you can actually in some ways predict who's going to respond based on their baseline epigenetic age. So yeah, again, like these can be used to actually pre-select your right population to do this on. I'm involved a little bit in so much as I've been on panels and stuff with Steve and other people. And I know, Nir's also worked a lot trying to get the FDA, not necessarily to look at epigenetic clocks, but to have aging surrogates as endpoints. So I've been part of the discussion, but yeah.
It seems like the work you described could be really useful because one of the big stumbling blocks to getting a clock used in a trial in terms of stratified analysis of efficacy is the expense of running the thing. And if, for example, one of the modules is really the main predictor, for example, of epigenetic reprogramming, maybe it's, maybe there's a way to derive cheaper but still effective ones that could be pushed for this purpose. Anyway.
Yeah, that's the other thing is what if we can actually map some of these modules or whatever they are to different outcomes or different mechanisms, you might not have to measure the whole clock. And I'm working with someone else to do more targeted approaches that hopefully you could actually get clocks that are $25 instead of $250, but they'd be more the singularly focused ones rather than kind of these more global ones. But yeah, I totally agree.
All right, thank you. Larry, you are up next.
By the way, Morgan has another meeting in 3 minutes.
Oh,
yeah.
okay.
Yeah,
So we both have to go.
what I was trying to get at, Morgan, is that it's really difficult sometimes to, separate out what the effect of methylation is. And when you're looking at chromosomes, is it forming condensation? Is it shutting down a lot of other things? And the sites that you're looking at, do you really know what the effect of methylation? Usually methylation leads to less gene expression and things like that. And are these genes, like if you look at senescent T cells, it's mostly, they express PD-1 and that makes them senescent for the most part. And so, is there ways to correlate this sort of stuff with, with the actual functionality that, this site, and, how do you look at aging? Is there a certain rate of methylation on a specific site that happens over a lifetime, or is that what you're really looking for? I mean, most of the time when you're looking at, at progenitor cells or things like that, there's not as much methylation. So are you just looking at sort of a site that has no function, but just gets sort of accumulatively methylated?
So the interesting thing that we're actually finding is it doesn't seem to be about specific sites or specific genes. It's more of these global patterns. And I think, yes, that's harder to put into, harder to contextualize in terms of mechanistically what it's capturing. I mean, it would have been great if it was just, you get methylation at a promoter region in this gene, which is decreasing transcription, and then that makes sense. It doesn't seem to be that. And actually, I could sub out a CpG into a completely different gene or completely different part of the genome that captures the exact same signal as this other one. So it doesn't seem to be something about the genes that they're controlling transcription for. So the answer is, I don't know what we're capturing. And I think that's kind of the motivation for this module thing, is if we can find CpGs that are tracking together, or at least seem to have these emergent properties together, then somehow we need to backtrack and map that onto mechanism. But yeah, it's very preliminary, and I think the answer is we just don't know right now what it is. Allison, you're muted.
Yeah, we have 1 minute left and you have to hop off, Craig. I think Keith had a comment more than a question. Do you just want to shoot it out there and then we'll close it down?
Super quick follow up to basically what Carl was saying. Is there being any thought applied to if a certain, based on which tissue samples you have available, how much compared to the whole Megillah you can get? So for example, if you can get like 90% accuracy from something easy to collect, maybe we can get a longitudinal sample much easier. So is there analysis ranking things like that, basically?
Yes. I don't think there is an analysis systematically ranking things like that, but that will be important going forward, right? Like, you have to weigh kind of how easy it is to get the sample, or the other thing is to weigh how expensive, right? So even with the original PhenoAge clock, even though we didn't publish this, it had 513 CpGs. You could get almost the exact same measure using half of that. So this will be important going forward if we develop these cheaper targeted ones that,
So I'd like to see,
yeah. But we should shift our thinking there. That's really important.
Morgan, thank you so much. We're now 1 minute above your next meeting, so we really don't want to hold you up, but thank you so much for joining. It was really fantastic. I know that at the end of this, there's always more and more questions clogging. Thank you so much for joining. I hope we can have you back in the near future when you're hopefully gonna have follow-up findings to share. Thank you very much from all of us, and I'll be following up.
Yeah,
and
great questions too. Thank you.
Bye, Morgan.